Dirty gloves straight from the pack. Are food handling gloves more hazardous than we imagined? If you're a food safety expert, you are no doubt aware of the many problems associated with disposable gloves in the food industry. Problems like gloves being worn for too long, pieces of broken gloves ending up in food, gloves being used in place of hand washing, skin, dexterity and comfort problems for workers, and the presence of dangerous chemicals like plasticizers in some gloves. For me, the biggest problem with glove use is the false sense of security workers can get when wearing gloves. Put on a pair of gloves and you can feel like you are clean, hygienic and contamination proof. And that can lead to workers making food safety errors such as handling cooked foods after raw foods without changing their gloves first. These are problems we know, and we've been talking about those in the food safety world for decades. What's new today is information published in July's issue of the Journal of Food Protection in an article titled Potential for Glove Risk Amplification via Direct Physical, Chemical and Microbiological Contamination. And that article is by Barry Michael and a bunch of other people. Barry Michaels and a bunch of others. It's a wide-ranging and frequently rambling collection of concerns about disposable gloves. Much of the article is unsurprising. For example, the authors discuss how supply issues created by COVID-19 created opportunities for unethical behaviour in glove supply chains, and share that when gloves fail, they release micro-rich sweat from the interior of the glove. For me, the surprising information in this article is about the microbial contamination present on gloves before they've been used. The authors share information from yet to be published research that says they found fecal indicator organisms on around 50% of samples of disposable gloves. The samples were collected from 26 brands of glove, which the authors say represent approximately 1/4 of the estimated 100 glove factories in Southeast Asia. The paper references other studies performed on non-sterile medical examination gloves and conveys the impression that many brands of gloves are teeming with pathogens. However, when I examined the original sources, it became clear that although microorganisms are present on new unused disposable gloves, the numbers are typically low. For example, Ferreira et al. 2011 found approximately 5 or 6 microbes and technically colony-forming units, or CFU, per finger. So that's 5 or 6 per finger on new disposable non-sterile latex medical examination gloves from a single brand. And Hughes et al. 2013 discovered 0.103 CFU per glove of environmental bacteria, less than 40 CFU per glove of skin bacteria, and no pathogens on latex medical examination gloves from newly opened boxes. Microorganisms on unused gloves from newly opened boxes are presumably present because of cross-contact with the environment during manufacturing or packing processes, or perhaps from contact with the cardboard used as primary packaging. Perhaps from contact with the cardboard used as primary packaging for most disposable gloves. However, packaging material was ruled out as a source of contamination by Michaels et al., who discarded the outer layer of gloves, which had been in contact with the box before performing their microbial tests. Instead, the source of microbial contamination of gloves is explained by Michaels et al. To be contaminated water used in the glove manufacturing process. Water is used to wash the formers, that's the moulds, which are dipped into liquid rubber or polymer to make the gloves. Water is also used to rinse newly formed gloves before vulcanizing in a process known as pre-leaching, and after vulcanizing in a process called leaching. Because gloves are turned inside out when they're removed from the formers, the surface that was in contact with the former becomes the food contact surface. Does this mean that a microbially contaminated former could be the source of microbes on the outer surfaces of gloves? Perhaps, or perhaps not, given that vulcanizing and leaching are high-temperature operations. The ability of microorganisms to survive the manufacturing process is not discussed in the paper. So, with evidence that non-sterile disposable gloves have some microorganisms on their surfaces, the important question is, do these microorganisms pose significant risk to food safety to food consumers? The authors of the new paper do not say. However, they do share stories of medical incidents caused by gloves, including one described by Sorio et al. 2023, in which new disposable medical examination gloves were found to be the source of bacteria which caused 139 infections in a hospital over 13 months. Another, described by Stock et al. In 2011, revealed that Aspergillus mold on non-sterile disposable medical exam gloves, which had been kept stored in their native packages, was the probable cause of the death of an infant from aspergillosis. Microorganisms found by the authors on unused disposable gloves include enterotoxigenic strains of Bacillus cereus and Bacillus anthracis, Listeria monocytogenes, Clostridioides difficile, Staphylococcus aureus, Pseudomonas aeruginosa, Streptococcus pneumonia. And most of these organisms are capable of causing foodborne illnesses. However, the numbers mentioned in this new paper are, as in previous studies, low. The authors state that their statistical analyses indicate, with some degree of confidence, that 10 to the 5 to 10 to the 6 CFU per 100 glove boxes could be possible. And that was a direct quote from the paper. So 10 to the 5 or 10 to the 6 CFU, that's up to 100,000 bacterial cells per 100 gloves, equivalent to up to 10,000 cells, that's 10 to the 4, per glove. My understanding is that these figures also include bacteria from the inside surface as well as the outside surface. Are these quantities of bacterial cells on a glove a cause for concern in food handling? Will such numbers cause food contamination that is severe enough to lead to illnesses? Perhaps, but I think it's unlikely. Have contaminated gloves caused outbreaks? None are mentioned in the new paper, and I wasn't able to find any outbreaks caused by microorganisms on fresh from the pack disposable gloves. A review of outbreaks caused by retail food establishments, and that was by Lipsky et al. 2019, noted that most outbreaks were from norovirus, a pathogen linked to hand hygiene failures, and less than 1/3 of establishments with outbreaks had a written policy on disposable glove use. But there was no suggestion that glove use was directly responsible for any outbreaks. Should we be worried? Is this yet another food safety hazard to add to our risk assessments? I don't think so. A small quantity of bacteria on the surface of a glove is unlikely to make anyone sick unless it gets into food that's subsequently subjected to what us food safety nerds like to call temperature abuse. And such food could cause an outbreak even if it had not been in contact with gloves. It's worth noting that most of the authors of the paper are either current or former employees of a glove company, Eagle Protect, and that the paper specifically mentions glove manufacturers in Southeast Asia on the topics of worker slavery, polluted water sources, and inadequate worker health standards. Takeaways for food professionals. Microbial tests on unused disposable gloves have discovered bacteria and fungi on the inner and outer surfaces of gloves at levels of up to 10 to the 4 CFU per glove. Organisms found on unused gloves have been linked to illnesses and deaths in clinical, that's hospital settings, but not to foodborne illness outbreaks. While the risk to food safety is probably low, it makes sense to avoid using gloves for processes that require extremely high levels of hygiene, and it's better to use a non-hand process such as tongs instead. It's also a good idea to source gloves from reputable manufacturers that are subject to audits and meet certification standards. Such manufacturers are more likely to supply gloves with lower levels of surface microorganisms. In short, disposable food handling gloves are non-sterile, and a new paper discusses the presence of bacteria on the surface of gloves. Bacteria and fungi on unused gloves have been linked to hospital infections, but not to outbreaks of foodborne illness. Consider replacing gloved hands with non-hand operations such as tongs for processes where hygiene is critical. Sourcing gloves from reputable manufacturers may help to reduce any risk from the presence of microorganisms on glove surfaces. And the main source for this post that I've just told you about is Michaels et al. 2024, their paper published in this month's Journal of Food Protection. And the other sources are referenced in line using hyperlinks. And as always, you can get those links from the email and the post.